Hello students, welcome to Vedantu NEET English. I am Ms. Gopika, your own biology master teacher. I hope I am audible and visible to all of you. Please let me know in the chats if I am audible and visible. And yes, we are going to start our class, right? We had a lot of problems, a lot of technical issues going around. But yes, finally we have made it. Just let me know if I am audible and visible quickly in the chats. Let me also just refresh and see. Yes, awesome. I am visible to all of you. Now tell me if I am audible as well. Yes, and we will start the class. Awesome. I'm audible, visible, everything is proper. Okay, let's directly get into the class. So what were we doing? We were doing molecular basis of inheritance, which is one of the biggest chapters of your, biology, and it is also considered one of the hardest chapter for most of the students because genetics is not like, okay, things like this keep happening, okay, and we cannot do anything about it, right? This is what it is. Okay, am I stuck by any chance? I feel it is hanging. Students, was there a lag or are we good to go? Okay. I think it's fine. Okay. Yes. So, before I go to the class, I want to inform all of you, students, last chance to fill the form, okay? Fill the form for what? The most voted cities. We 4 teachers, that is Diksha Ma'am, Baswa Sir, Capto and myself, we will be coming to your city, okay? So, we will be coming there, we will be helping you with your counseling, we will be helping you with what is your future plan, 2026 roadmap, 2027 roadmap. All these roadmaps will be given there. With that, you are also going to get some, free study materials as well for your NEET 2026. So I want all of you to come because we are traveling all the way from here to your city. So I want all of you to make it. Now, which are the cities that you can, fill the form for? Like this is the video that you're watching. Just go down and you will see fill the form. Click on this and you will be able to see. You have to enter your name, contact number and the city preferences. Okay. Right now, we are on lead. And do you want me to reveal the place? So we are on lead with Chennai and Hyderabad. Now there might be a tie, so I want you all to do the tie breaker. Okay, so if you're from Chennai, quickly go and fill the form and do the tie breaking so we can come to your city first. And if you're from Hyderabad, also do the same. And whoever can travel in an hour or 1.5 hours in and around Hyderabad also, please do come there. Okay, please do come there because we would love to see our YouTube students. It's the first time we are coming to Hyderabad. We have been to Chennai multiple times. So I want to meet all, all my Telugites, okay, all the Telugu students, I want to meet you all. So make sure that you fill the form today itself. Today I think by evening, we are going to close the form. Yesterday itself, the form was closed. Then so many students texted us on Instagram telling, ma'am, we sorry, we forgot about the form. So please fill the form by today, okay? We want to break the tie. Clear? I want all of you to do that much for us because we are working day and night for you and I think you can fill a form and come and meet us, right? Awesome students, please keep chatting in the, please let me know if everything is proper because I do not want one more technical issue to come. Okay, and quickly smash the like button, yaar. Why you people so conjuice? Why you people so conjuice? I don't understand. Yes, okay, let's start. So last class we understood up to packaging of DNA, students. So packaging of DNA, beautiful notes I have sent all of you on the Telegram channel. So you can download that notes and you can revise whenever you want. So today we are going to start with search for genetic material. See, even though, people were telling, okay, like DNA is the genetic material wasn't told to us until very late, okay. DNA is the genetic material. Actually, this search, no students, that DNA is the genetic material went on for a very long time and only in 1926, okay, 1926 we got to know that, okay, DNA is the genetic material or the quest means the hunger to find it. Now you have a hunger, no, to find if your friend went for this party or not, like that hunger was there among the, zoologists, botanists that, okay, we should actually find if DNA is the genetic material. But see, other people also helped us, maybe give us a little bit about your, about the DNA. Okay, I think everything is fine. Okay. Yes, I want continuous chats to be moving and I want continuous smashing of like button and one person to put on the target as well. Okay, target as well. Okay. So students, see here, according to your principles of inheritance, which sir is going to do very soon for you, Sutton and Boveri, Mendel Morgan, all these three people gave us one thing, that is, there is nucleus and inside the nucleus there is chromosomes, right? This is what they gave us ideology from the starting, that guys, this is the cell and inside the cell there is going to be a smaller structure. Oh, this is also moving. Okay, there is going to be a small structure which is called the nucleus. Okay, we'll draw it the small nucleus, and inside the nucleus they told us there is a presence of a chromosome. Yes or no? But did they say us that genetic material is DNA? No ma'am, they did not tell us. Now isn't it very important for us to know DNA is the genetic material? Yes or no? Tell me quickly. Yes, okay. Okay, DNA is the genetic material. We got to know? Yeah, we got to know very, very recently. Okay, so what did they give us ideology? That yes, there is nucleus and inside the nucleus there is chromosome. This is what they told us. Okay, so now we will understand exactly how did they come to a conclusion that DNA is the genetic material. So I'll teach you very slow. Okay, I'll teach you very slow so you understand. I know some of you are studying for the first time. Don't get overwhelmed. I'll teach you very, very slow. Okay, now let's start with the first thing. Okay, there's a paragraph given in NCERT which is one of the very important paragraphs. Even though the discovery of nuclein, I remember I taught you about nuclein, Friedrich Mischer, when he was working on pus cells by fluke, right? I told you, by fluke, when you're going to search, let's say you're going to search something else, like you're going to search coriander, by mistake you got curry leaf and luckily your mom asked for curry leaf. That means what? By fluke you're getting that. Okay, so nuclein by Mischer. Okay, nuclein by Mischer, and the proposition for the principles of inheritance by Mendel. So here what are they talking about? I know most of you haven't studied principles of inheritance. Here they're talking about the pea plant, right? He took round, wrinkled, yellow, all that he took, no? So Mendel was the person who told that there is some factor. Okay, he did not call it gene, he called it a factor that was deciding the physical trait, or that was deciding the genotype and the phenotype. So Mendel also introduced us to chromosome, introduced us to, allele and factor. The quest to determine the mechanism for genetic inheritance had reached the molecular level. That means people were like, I am really hungry to find who is this cell who has all the information and who is this person who is helping in all this transfers. Okay, transfers. So previous discoveries by Gregor Mendel. So that's what I wrote, Mendel. Okay, Sutton, Morgan, and numerous other scientists had narrowed the search to chromosomes, right? Chromosomes located in the nucleus of most of the cells. But the question of what molecule was actually the genetic material had not been answered. So are you all ready to do the first experiment, that is your Griffith's experiment? A beautiful experiment, okay? A beautiful experiment. Is it clear? Yes, okay. So here students, what was happening? So Griffith was working on, okay, is there a lag? I think there's no lag, okay? I think it was like, maybe for a second or so there's a lag, but otherwise it's fine, okay? So Griffith was working on, important, okay? What was he working on? He was working on Streptococcus pneumoniae. What is he working on? Streptococcus pneumoniae. Students, from this word, what and all do you understand? Ma'am, I understand that coccus means they are circular, right? So whenever you see some word, no, you make sure that you decode it with the other chapters as well. All of you remember coccus means they are spherical in shape. Coccus means they are spherical in shape because we studied this in biological classification. From the word pneumonia, what do you understand? Ma'am, they are causing, okay, they are the causative agents for pneumonia. So this much you can, decode. Eureka you can say, okay, eureka. Pneumonia, correct. This much we understood? Yes ma'am, understood. Now, when he was working on Streptococcus pneumoniae, these people form colonies, right? And these colonies had formed into two different, okay, one S strain and one R strain, okay? So when he was working, he started noticing. So let's draw a Petri plate. All of you know how a Petri plate looks like. So this is the Petri plate. In one of the Petri plate, he saw that there was one different strain, and in one of the Petri plate, he saw a different strain, okay? So in one, he saw some very nice smooth colonies. What did he see, students? He saw some smooth colonies. Smooth colonies means what? That means something like polished, right? Polished, like proper capsules are there, smooth colonies. And then in one he saw some rough colonies, okay, some irregularly shaped, some rough colonies he saw. So he noticed that, okay, these people are two different strains, okay. So here he went on understanding that Streptococcus pneumoniae, the first that I saw was a smooth strain. So I can say, students, in further I will be writing S strain. Okay, when I say write S strain, it should be understood that I am talking about smooth strain. And here he noticed that these people were little irregularly shaped. So these people are the rough strain. Okay, these people are the rough strain. And what will I call it? The R strain. Okay, I'll call it the R strain. Understood, all of you? Please put thumbs up as you understand. So smooth strain, which is the S strain, R strain, which is the rough strain, which is the R strain. Okay. Now there was not ending to this. Okay. He took it. He wanted to know more about these people. So he took to just look at it. How do they look? What is making them the smooth strain? We should understand, right? So he took, and we will draw a little larger image. So student, this was the smooth strain. Okay, one circle I am drawing, and out of this layer I am drawing one more circle. Okay. Okay. Yes. Okay. Can you see the pink and orange? It's kind of visible, but not very dominatingly visible. So he looked at the smooth strain and he noticed that I want it to be pink. So we'll do one thing. Okay. We have ideology for everything because idea Vodafone. Okay. So students, here what happened? He looked at this and he noticed that these people have an outer coating. Okay. Outer coating. This outer coating was what? He called this outer, he noticed that this outer coating was made up of a mucopolysaccharide, okay? Mucopolysaccharide. In some books, you will also see in simple words, they say there was a covering of a capsule, okay? There was just one capsule. You can see in some books saying that, okay, the smooth strain had one capsule cover to it and that's why it was called the smooth strain, okay? So this is your S strain. And then some people also told that this mucopolysaccharide, right? Excuse me. Okay, this mucopolysaccharide was the reason why your S strain, okay? My ears got it. Okay, this S strain became virulent. Virulent means what, students? Disease causing, okay? Virulent means what? Disease causing. Shall I write it? Disease causing. This board is too tiny, so I cannot fit in everything here. This much is clear? This much is clear? Yes or no, quickly in the chats? Okay, clear, right? Quickly mark your attendance. Quickly mark your attendance. Done. Okay. G, ma'am. Okay. Now, this much is done, that there is a mucopolysaccharide covering on this S strain. That is why this gave a smooth appearance. Okay. That is the reason why this gave a smooth appearance. Students, you know that, what is that? What do you call that? A quote saying that never fall for an appearance. So that is this fellow. He had a smooth appearance, he had proper capsule layering, but what was the problem? The problem was that he is virulent, okay? But this Pavam R strain is there, no? On the other side, he is one innocent man, okay? He is one innocent man. Why I'll tell you? Because his covering, okay, his coating was something like this. Oh no. His coating was something like this. Completely disoriented kind of a coating. Okay, this kind of a coating. He did not have any capsules outside. He just had this one layer. So this person was the R strain. And what was the important thing about the R strain? There was no mucopolysaccharide coating. There was no mucopolysaccharide coating. strain, we understood one thing, this is not virulent, okay, it is non-virulent. That means it does not cause any disease, okay, it is not disease causing, not disease causing. Clear to all of you? So, very simple. What happened? Griffith was working on Streptococcus pneumoniae, which causes pneumonia. Now, he noticed that when this growing of Streptococcus pneumoniae happened, he saw two different colonies. One was S strain. That means different strains are there for all the bacteria, right? So, he noticed that one was a smooth strain and one was an R strain. So, one was smooth because of the mucopolysaccharide coating and one was R strain, that is a rough strain without any mucopolysaccharide coating. Okay. When he looked at both of them, he noticed that S strain was virulent. How? By injecting into the mice, but I am giving you an introduction. And when he injected the R strain into the mice was alive, making us come to a conclusion that smooth strain is disease causing and R strain is not disease causing. Okay, R strain is not disease causing. So, now this much we understood, not disease causing. Now we will try to understand the experiment. Okay, students, all this happened in 1926, so do not forget the dates as well. Okay, so what was happening? He took mice, okay, every time students, okay, mice is the only one who is you know, being sacrificed. Okay, so he took mice, okay, one mice, two, okay. Now, what did he do first? First, he injected it with the S strain. First, he did what? He injected it with the S strain. What would have happened? All of you tell me in the chat box quickly. He injected it with the S strain. What do you expect to happen? Faster, faster in the chat box. I'm waiting for your answers. What would have happened if he injected it into the mice? Quickly. I'll see the chats here. I think here it is a little faster. Yes. All of you did not mark your attendance. Okay, no Kirti, I am from Bangalore but I didn't go there. Where is that? Okay, so look here, S strain, it was understood that the mice would die, right? The mice died. Because why? We know S strain is virulent, it is disease causing, so the mice is going to die. Okay, very good. And see, now here what happened? Here what happened? He injected it with the R strain. He injected it with the R strain. What do you think here it would have happened? What would have happened here, students? Ma'am, I understand that here the mice would have, mice is alive, okay? The mice is alive. Now then came, he was like, okay, wait, why don't I take it to a next level? I will take the mice again, okay? I'll take the mice again. Okay, you should give me for this diagram, I'm telling you. This diagram is like look at the mice. Can you draw a better mice? Okay, so what did he do? First he did heat killed S strain. That means what? He gave extreme heat. Okay, he heat killed the S strain. But students, you know DNA, right? DNA is chemically, structurally stable and it is not going to be completely damaged by your heat. Okay, but when we talk about transformation, I'll tell you what, why, you know, reason, how did this transformation happen, I'll explain. So what did he do? He took heat killed. He took heat killed S strain. He took heat killed S strain and then he injected into the mice. And for his, you know, he was very fortunate and he noticed that the mice is alive. Okay, or mice lives. Okay, mice lives. Done. Then what happened? Then what happened? Then he was like, why don't I take it to a next level? Okay, why don't I take it to a next level? I take two positive results. Which are the two positive results here? My two positive results are mice is alive, mice is alive. These two are my positive results. So he was thinking, why don't I make a combination out of them? And he simply took heat killed S strain. Questions come from here only, okay? He took heat killed S strain plus R strain and he injected into the mice. Students, if you were Griffith, okay, if you were Griffith, what would you expect? What would you expect if you were Griffith? Heat killed S strain plus R strain. You would have expected that mice would be alive because in both the scenarios, mice was alive. But here comes the S strain to the R strain that made R strain virulent. This he was sure. He was sure that something has happened, that when I combined this combination, it led to something getting transferred from heat-killed S strain to R strain, making R strain virulent. That means what? Making R strain the villain. So you sit with a villain for a very long time, what happens? Slowly you will also start thinking that I am the villain, right? Because he's giving you continuous negative energy, telling that you should do this, you should do this, stealing is okay, cheating is okay. So you will also start being like, oh yeah, it's okay, like he's surviving happily, why don't I also do that? So influence, okay, bad influence, right? So here what happened? Mice died, okay, mice died. So now comes the actual, what exactly happened. So see, there were, conversations that S strain is virulent because of the mucopolysaccharide. Now here what happens, students? In the heat-killed, okay, heat-killed S strain, even after heating so much, the genetic material is there, right? So there is a process called as transformation. Okay, the genetic material wouldn't have been completely destroyed, it would be active. Now this transformation process is where, naked DNA. Okay, now we do not know what is the genetic material, so we will just say naked genetic material. Now you and me know that it is DNA, but that time they didn't know about it. Okay, so let's be also a little like, low key. So naked genetic material is there. It is normal in bacteria, even in sexual reproduction. I taught you in biological classification that they do transformation. That means what? From the environment, if there is a naked genetic material, these people will pick it up. So here exactly what happened? This was picked up by the R strain. This was picked up by the R strain, and we call this process as transformation. We call this process as what? Transformation. So do you think Griffith understood what went wrong? Griffith knew that something is happening, something is transferring, but Griffith did not know what was transferring. So he simply called this thing that got transformed, okay? He told that something got transformed from the heat-killed S strain to the R strain, and he simply called it the transforming principle. He called it what? The transforming principle. Understood all of you transforming principle experiment? Quickly let me know in the chats. Did you all understand transforming principle experiment? Hi Lisa. Yes. So he called it transforming principle. Students, did he tell what was the thing that got transferred? No. Did he tell that the mucopolysaccharide is the reason? No. He was like, something has got transformed from the heat-killed S strain to the R strain, making R strain virulent. Making R strain virulent, leading to what? Leading to death of the mice. Leading to the death of the mice, and he called it the transforming principle. So he did not work on the biochemical nature, okay? Griffith. Many students, get confused with it. Did not work on the biochemical nature. Biochemical nature of heat-killed S strain. He did not work, okay? He did not work on the biochemical nature of heat-killed S strain. So someone has to do it, right? If I don't do it, hi Jerisha, sorry, I keep getting confused. Okay, if I don't do it, someone else will do it, right? It is very normal. If I do not do it, someone else will do it. So this is what happened. This is what happened. Done. Transforming principle is clear. So what happened? Quick recap, okay? Quick recap. Come on, all of you keep answering with me, so it will be better, okay? So what was he working on, and when did this all happen? In 1926, okay? He was working on Streptococcus pneumoniae, and he noticed that there were two colonies that was formed. One was the S strain and one was the R strain. S strain is called the smooth strain because it had a smooth appearance because of the presence of a mucopolysaccharide, or in simple words, you can say a capsule layering, which made the smooth strain strain virulent, okay. And then students, when we notice the R strain, we notice that the R strain, okay, R strain did not have any mucopolysaccharide covering, no capsule covering, nothing was there. And it looks very sad, but then it was non-virulent. That means it does not cause any disease, okay. Now, what did Griffith do? He started doing experiment with the mice. First, he took the mice, he injected it with the S strain, and he noticed that the mice is going to die. It was like an obvious answer. If something is causing us disease, we know that if we inject it into them, they are going to die. Second thing, mouse plus the R strain, we knew that this fellow will not kill the mice, so the mice was alive. Now, twist and turns started happening from here. He took mice, he heat killed the S strain. That means after giving extreme heat, he heat killed the S strain and the mice was alive, okay. And then he was like, okay, let me take both of my positive results and do a combination. He took mice, heat killed S strain plus R strain, the mice died, okay. Thank you so much, Naz. Shagufa Naz, okay. I think I'm reading your name a little wrong, okay. Thank you so much, dear. Okay, now students, heat kill. So, one thing Griffith was confirmed, that something has got transformed from the heat killed S strain to the R strain that made the R strain virulent. This he was sure, okay, but he did not know exactly what, okay. Silent people, yes. So, heat killed S strain, some naked genetic material, right? You know that the entire naked genetic material is not going to be destroyed. So, this naked genetic material was picked up by your R strain, and that process is called transformation, which I have taught you in biological classification, okay. So, he called this transformation as the transforming principle, which led to making the R strain virulent, leading to the death of the mice, okay. Now, Griffith did not work on the biochemical nature of the S strain because he was like, I am not going to go further with it. I am happy with this heat killed, S strain story, right. He was just only bothered about that. Then, another people came. They were like, no, we have to go forward and find it out. So, that is where the biochemical characteristics of transforming principle, and students, you look at the lifespan, almost 10 years. 10 years they were trying to work on it and still they did not become successful because it was not the most accepted one. Now also it is not the widely accepted one. Clear to all of you? And McCarthy, okay. All of them sound like, when McDonald's meal, but they are not, okay. So, Avery, McLeod and McCarthy. I think the C is capital, no? These three people were like, wait a minute, I want to work on, okay, I want to work on the transforming principle, almost 33, hey, this 11 years, yeah. So, almost they were like, no, we want to work on and understand what exactly got transformed, okay. They were very sure that something has got transformed and they wanted to make sure that who or what was the thing that was transformed, okay. So, students, first of all, the entire setup I am telling you, okay. Their entire setup was mainly focused on enzymes, okay. Their main focus was enzymes and they took 3 enzymes into consideration. First one, because see, when you are thinking about biochemical nature, what are the doubts that you have? Let's say you are a scientist and you are thinking. Yes, tell me, what was the Hi Manos, yeah, hi Nas, Science World, okay. Yes, so look here, I want Science World kind of energy, Jerusha kind of energy, okay. I do not want this boring, sleepy people in the class. Come on, students, we have to go an entire year, and if you're going to be like this, it's going to be very difficult, okay. Yeah, now look here, 3 enzymes. Now, you only just imagine, you are working on this. What and all doubts will you have? Ma'am, I'll have doubt on protein, I'll have doubt on RNA, I'll have doubt on DNA, right? These people also had the doubt. So, first they took enzyme called as protease, okay. Next, they took enzyme RNase. Then they took enzyme DNase, okay, DNase. Now, if you look at protease, RNase, DNase, you notice that this word, no, ACE, okay. This means these people are degrading enzymes, okay. These people are going to help in degrading. So, this protease will degrade protein. Protease will do what, students? It will degrade protein. RNAs will do what? Degrade RNA. What will DNAs do? Ma'am, understood ma'am. Degrade, okay. If my students were now offline, they are told, ma'am, understood ma'am. Degrade RNA. Okay, so this was their main setup. They took this, and the second thing that they took into consideration was what, students? They took the heat killed strain. Okay, heat killed strain. Okay, that is the biochemical constituents. Constituents. Okay, yeah. So this was their main setup, that I will work on this. Okay, yes, got it. Done. Okay, degrade RNA, very good, awesome. So Avery, MacLeod and McCarthy was working on this from 1933 to 1944. And what did they do? Now the experimental setup is very important. Okay, we will take a test tube, or we can even take a box and understand. Okay, I'll take circle first. Okay, 1 circle, 2 and 3. Now I want all of you to listen. This experiment can be a little confusing, but we will try and understand. Okay, we will try and understand peacefully. Okay, yes. Oh, SR gamer, don't spam. Please be calm. Okay, please be calm. Spam mat kar do. Yes. Students, quickly mark your attendance. Come on, quickly. There are many students who have not marked their attendance. I know for sure. Okay, I can tell exact numbers. Come on. Quickly smash the like button. I need some energy, yaar. Come on. Yes. Okay, done. Now look here. Let's take protein in green color. Okay, you're going to remember the colors. This is protein. Okay. Let's take RNA in red color. No need red, actually I need to cancel it out. Okay. Let's take pink color for RNA, and let's take blue color for DNA. Okay, I'm just drawing it just to color indication. Okay, color indication. I want all of you to remember the color. Green is for what? Green is for protein, pink is for RNA, and blue is for DNA. Okay, just to make you understand better. So now just look at this. 3 setups I have taken. Okay, 3 setups I have taken. Now in this 3 setups, you have to remember one thing, that these people are going to take the enzymes, right? Now students, I have seen 2-3 different way of teaching this, where they take protease plus DNase, RNase plus DNase. So they do a combination where they want to show there is no transformation. But I feel this way is easier, because this experiment does not have complete clarity in your NCERT. Even if you search a lot about this experiment also, there is no full clarity. It is kind of little confusing. So to make you understand the paragraph of NCERT, I would like to teach it this way. Okay, I would like to teach it this way. So look here. Here first they wanted to know who is responsible for the transforming principle. That was the main goal. First, students, the enzyme that they took was protease. They first used protease. Now you tell me, if I take protease, right? If I take protease, let's just copy paste it here. 1 second. Yeah. So if I take protease, what is going to happen? All of you tell me in the chat box. If I take protease, then, okay, yes, this experiment is a full confusion. Today that confusion will go, okay. If I take protease, it is very sure that this protease is going to degrade my protein. Yes, ma'am, it is going to degrade the protein. Correct. That is the job of this fellow, to degrade the protein. So he is going to degrade protein. Now you tell me one thing. If your protein is degraded, does this still kill the mice? I would say yes, it is going to kill the mice. Why? Because DNA is still active, right? DNA is still active. Now, look here. It will degrade protein. Then it is understood that the DNA is still going to get transformed through the astin. So here students, they say that no transformation happened, or I would say the mice is still going to die, right? Mice is still going to die. Why? Because our DNA is still active, right? DNA is still active, so the mice is going to die. So I would say transformation will happen, okay? Transformation will happen. Yes or no? Will transformation not happen? Yes, ma'am, transformation is going to happen, right? Right? Transformation is going to happen. So look here, DNA is still active here. Once again, I am explaining you people. Protein, RNA, DNA. Okay, protein, RNA, DNA is there. First, I added protease. It is going to degrade the protein. If it degrades the protein, my protein is not going to be there. In the combination, in the biochemical nature, protein is not going to be there. But RNA and DNA is there. DNA is the one who is going to get transformed. That means if I add R strain, this fellow is still going to transform. Transformation will happen. Yes, ma'am, transformation will happen because DNA is still there. DNA is still there and DNA is going to get transformed and the mice is still going to die. Simple way of understanding. Are you all understanding this much? If you understand the first one, then it is easy for me to explain the rest of it. So please let me know if you are understanding quickly in the chats. Quickly in the chats. Fast. Just tell me if you understood this much or not. Who is that who is asking the full form at this point of time? Huh? Is this the time for you to ask the full form of RNA? Tell me. Please let me know if you understood. I am waiting for your chats to move. Waiting for your chats to move. Yes, ma'am. All of you? Because this is a little confusing. That's why I want to make sure that you have understood it. Done? Awesome. Okay? Yes. One more time. So look here. Protease is there. It is going to degrade the protein. Protein is going to degrade and transformation will still happen. Why? Because protein is not the genetic material. Right? Simple thing. Because protein is not the genetic material. Understood? Now you will be able to say for RNA what happens. RNA, what is going to happen? I am going to take RNAs here. I am using the same color so you understand. I am going to take RNAs. If I take RNAs, what is going to happen, students? If I am going to take RNAs, it is going to degrade my RNA. Yes, ma'am, it is going to degrade your RNA, right? It is going to degrade your RNA. And what is going to happen? Here it is very simple that here also transformation is going to happen because DNA is still active. Transformation will happen. Okay? Transformation will happen. Why? Simple reason is that because RNA is not the genetic material. RNA is not GM. GM means genetic material, okay? RNA is not the genetic material. Done. This is also clear? Yes, very good. Now last one, we have our DNAs. Last one is going to be our DNAs. So here RNAs is going to degrade. RNAs is going to degrade RNA. And DNAs is also going to degrade DNA. Degrade DNA. Okay, let's copy this also here. Here, what is going to happen, students? Ma'am, here your DNA is finally going to get degraded. And if your DNA gets degraded, okay, if your DNA gets degraded, that means transformation will happen or not? Ma'am, here transformation will not happen. No transformation. Why no transformation? Because DNA is the genetic material, and if DNA is not there, then who is going to get transformed? Okay? At least know my name, dear. Who is that? Napoleon. At least you should know my name completely. Okay? Done. Excellent. Excellent. Okay? Yes. So here what is happening? DNAs degrade DNA. DNA is gone. Now you tell me, will transformation happen or not? No transformation. Why? Because DNA is the genetic material. DNA is the genetic material. Genetic material. This is the thing. But this experiment had something missing. This experiment had something missing, because of which many people were not okay with it. Many people were like, no, it's a lot of confusion, there's no clarity in the experiment, and they did not approve this experiment. What happened, students? They did not approve this experiment. In other words, you can think, see, look here, if protein is degraded, here still the mice is going to die. Why? Because DNA is going to get transformed. Here also same thing. If RNase is used also, here also mice is going to die. Why? Because again DNA is going to get transformed. But here if I use DNase and degrade the DNA, the mice is going to be alive. Why? Because transformation is not going to happen. So I teach in two ways. Okay? I teach in two ways. Clear? So here transformation will happen, no transformation, because I destroyed the DNA that was supposed to get transformed from the heat-killed strain to the R strain. This was Avery, MacLeod and McCarty's experiment. Any doubts? Any doubts? Thank you, Vansh. Any doubts so far? Please let me know. This is very important concept and many students do not understand this clearly. So that is why I am asking, multiple times. Any doubts here? Please let me know in the chats. I am ready to explain 100 times to all of you. Okay, we will do a quick recap. First, you just give me a sign that you all understood. No doubts? Okay, no doubts? Awesome. Okay, we are very close to the target. Come on. Ma'am, once more. Okay, sure. Let's do it. Students, this is the best part. If you start the syllabus early, we have a lot of time. Okay, but if you start the syllabus very late, then we do not have time to explain this many times. We will be in a hurry. Okay, look here. Avery, MacLeod and McCarty. These three people. See, after Griffith told that this is the transforming principle, Griffith left that experiment. Griffith was like, it's fine. Okay, Frederick Griffith. Okay, full name. Frederick Griffith. He was like, it's okay. I do not want to continue with this experiment. Then Avery, MacLeod and McCarty came. And what was their main strategy? They understood that DNA is the genetic material, but they had doubt. Is it protein? Is it RNA? Is it DNA? They had slightly doubt. Okay. So they took enzymes called protease, RNase and DNase. Protease would degrade protein. RNase would degrade RNA. DNase would degrade DNA. Okay. Then he took the heat-killed strain. That means they took the biochemical constituents. What is the biochemical constituents? Protein, RNA and DNA. Okay. Then what setup did they do, students? They took test tubes. Okay. They added the protein, DNA, RNA. I'm just showing the setup. And first they used protease. ACE means ASC, enzymes that would degrade. Okay, you can remember like that. Protease degraded the protein. Protease would degrade the protein, but will transformation get affected? No, ma'am, because I already know DNA is the genetic material. So here what happened? Protein got degraded and we know that protein is not the genetic material. So transformation is going to happen. Right, students? Transformation will happen. Transformation will happen means what? Mice can still die because heat-killed strain still has the genetic material and that will go get transferred, right? Transformed. Now, second scenario, again same setup, we use RNase here. We used RNase. So remember here, okay, they might have added protease plus RNase. It doesn't matter. Protein is already degraded. We degrade the RNA. After degrading the RNA, we again understood that RNA is not the genetic material. So still transformation will happen. Why? Because DNA fellow is still active. He's like, I want to go. Okay, I want to go. So next we have DNase. Last option was DNase. That means the one that would degrade the DNA. So DNase we took, it would degrade the DNA. DNA got degraded and no transformation. Why no transformation? If DNA itself is not there, then nothing is going to get transferred from heat-killed S strain to R strain. So it was understood, okay, it was understood that DNA is the genetic material. But like you people only, even many scientists, botanists, zoologists were like, this experiment does not give a clarity. So they went for another experiment. Okay, they went for another experiment. Okay, all the notes will be available on Vedantu NEET English Telegram channel and Gopika Biology Official. These two Telegram channels you can join and you will get the notes. Done? If you want to stay connected with me, you can join my Telegram channel, and if you want all the notes of PCB, you can join Vedantu NEET English Telegram. Okay? Yes, Jaypal, I hope it is clear. Now, can we go to the next experiment, students? Hershey and Chase experiment. Quickly give me a positive sign. Give me a positive sign that yes, ma'am. Jaypal, I already answered your chumma spamming. Chumma is simply, okay, in my language. Okay. Now, Hershey and Chase experiment in 1952. Students, do you want to see NCERT ones? Okay, do you want to see NCERT ones? I do not have NCERT here, so we will just search here directly. Okay, I do not have NCERT snips with me. So we can, oh, not notes. Okay, this is your new edition NCERT. Okay, how do you know it's your new edition? It will be written reprinted 25-26. So whenever you are opening, you open and check that. Okay, all this we understood. Okay, histone octamer we understood. Yeah, see, search of genetic material, from here only it starts. Okay, transformation principle in 1926 the quest started. 1928 Frederick Griffith's experiment started. Okay, so in a series of experiments with Streptococcus pneumoniae, bacterium responsible for pneumonia witnessed a miraculous transformation. Okay, so here rough colonies were there and smooth colonies were there, so we call it rough S strain bacteria and R strain bacteria. Okay, and this is what happened. And see, I just want you to read this one. Okay, I want you to read this one. See, biochemical characteristics of transforming principle prior to the work of Oswald Avery, MacLeod and McCarty. In 1933 to 44, the genetic material was thought to be a protein. So understood, many people thought that the genetic material was a protein. Okay, brightness is too much. Huh? 1 second, I'll reduce. Better? Yeah. They worked to determine the biochemical nature of the transforming principle. They purified biochemicals like what? Proteins, RNA, DNA, etc. from the heat-killed S strain. Okay, to see which ones could transform the live R strain into the S cells. They discovered that DNA alone from S bacteria caused R bacteria to become transformed. They also discovered that protein digesting enzyme called protease and RNA digesting enzyme called RNase did not affect the transformation. So, the transforming substance was not a protein or RNA. Digestion with DNase did inhibit. Inhibit means what? Stop. Okay, inhibit means stop. Okay, transformation. Suggesting that DNA caused the transformation, they concluded that DNA is the hereditary material, but not all biologists were convinced. Can you think of any difference between DNAs and DNAs? Sorry, DNA and DNAs. Okay, here in this chapter there is no change, dear. In this chapter there is no change. Students, what is very simple here? DNA and DNAs. What is the difference? What is the difference? Okay, only one A is there. I'll make you write it there. So, is this clear? Now, genetic material is DNA. We are going to the next experiment. Okay, we are going to the next experiment. I forgot to add NCERT snip. That is why I thought, okay, I will just tell you people. Nothing. Only one difference is there. No, what is the difference? DNA is the genetic material. Sometimes, you know, NCERT line questions will come. Like in-text questions we tell, no? DNA is the genetic material and DNase is the enzyme that degrades the genetic material. Okay, it's the enzyme that degrade DNA. Okay, that's it. Simple, one line. Okay, yes. Now we will start with this Hershey and Chase experiment. Ready? No difference in molecular basis of inheritance, so don't worry. No difference in molecular basis of inheritance. Okay, now Hershey and Chase experiment also, I want all of you to be very mindful what I am telling, because little bit confusions can be there, but we will try as much as possible to break it down. Okay, so Hershey and Chase experiment happened in 1952. Remembering that, let me just write here Frederick measures also. It's 1928. Okay, 1928. Here also let's make the correction. It's 1928. Okay, it's seen, right? Yeah. Okay, here. Okay, done. Now, coming back here, Hershey and Chase experiment. Okay, Hershey and Chase experiment happened in 1952, students. Okay, 1952. First, we learn about their setup and then we will come into the experiment. Okay, because it's very important. You know what are the things that they have used. So, first they used a bacteriophage. Now, you should know what is a bacteriophage. I have already taught you in your, biological classification. But still, in case you forgot about it, let's just learn it. So, this is a bacteriophage. Okay, inside this, there is going to be the genetic material. Okay, inside this, there is going to be a genetic material. Now, you should know what is this outside coat. What is this? It is the protein coat. It is what? The protein coat. Remember, it is made up of capsomeres and all that I taught you. Now, this is the tail. Okay, it is the tail fiber, which will help in attacking or attaching to the bacteria. Okay, which will help in attaching to the bacteria. Done? Okay, which will help in attaching to the bacteria. Now listen here. Inside this, what do you have? Ma'am, we have the genetic material. Now here it can be RNA or DNA. Here we are talking about DNA. Okay, here we are talking about DNA. Genetic material. This is the structure. Now what is this? What did I tell you? This is a bacteriophage. Bacteriophage. Now what is a bacteriophage? It is a virus. Important, okay? It's a virus that infects bacteria. What is it? It's a virus that infects bacteria. Now students, you already know virus is only active. Right, it becomes only living when it meets a host, right? Living when it infects a host, right? When it infects a host. Without a host, your virus is not very active. Okay, it is like a non-living thing. So here you have to understand that when I say about, virus, you have to understand we are talking about virus that is living or active when it comes in contact with a host. Why? Because it will release the genetic material into the host, right? Now, other than that, what were the things that they took? They basically took 2 radioactive substances, okay? Important for you to know. So, in one, they took radioactive sulfur. Now students, radioactive sulfur they took because, see here Hershey and Chase experiment was based on only 2 things. They wanted to know if it was a protein or if it was a DNA. They never had RNA in the mind also. Understood? Their main goal was protein or DNA. That's it. They didn't have any other thought. They didn't have any other thought. Their only thought was like, okay, it might be a protein or it might be a DNA. Got it? Now, first they took radioactive sulfur. Now, remember this very importantly. Sulfur is only present in protein and not, okay, they are like best friends. And since they are best friends, they both went to a shop and they saw this one white cloth top. Okay, it was very pretty. Both of them wanted it. Now what Jerusha will do? Jerusha will take her kajal and put a dot on the white top. Now even though the shopkeeper is going to hit them, that is a different story. Now what did Jerusha do? Jerusha did labeling. Labeling means what? She knows that, okay, if this black spot is there, Supriya is not going to take the stop, so I can take the stop later. So she did labeling. Now it was one of the traits of Jerusha that whenever she likes the top, she puts a black dot. Now I consider that as one of the labelings. Now on the other hand, Supriya, what she does, she makes sure that she wears a lipstick or lip balm, she takes and put a dot on that. So now we understand the difference, right? Black dot is seen, it is Jerusha's dot, and pink dot is seen, it is Supriya's dot. Okay, or let's say Chintu Mindu's, you know. Now this is called as proper differentiate labeling, right? I know who is labeling who. To make sure that protein labeling is separate and DNA labeling is separate, I took two substances. One was radioactive sulfur. I did not take, Hershey and Chase took, okay? Radioactive sulfur, which is only present in a protein and not in DNA. Understood? So they took sulfur 35. Oh, they took S35. Okay, they took S35. Now, this experiment did not stop here. Okay, we had to continue. So here, second scenario, what happened, students? Second scenario, what happened? They took, second setup, they took radioactive phosphorus. Now, where do you think radioactive phosphorus is going to be there? And what do you think it is going to label? Radioactivity is for labeling properly. Okay, radioactivity is for labeling properly. So far, did you understand? So, radioactive phosphorus, students, one catch about these people, they are only, phosphorus is only present in DNA and not in proteins. So this would only label DNA. It would not even be somewhere near to the protein. Understood? So this is specific labeling. So they took P32. Okay, they took P32 radioactive. Why we do radioactivity is so that we know this is one way we do labeling, because we cannot take a sketch pen and start labeling, no? So we take radioactive substance so we can do the labeling properly. Done? Yes, very good. Radioactivity only present in phosphorus and it is not present in protein. Good job. Okay, one second. Yeah. Now continue. Let's continue. So this is done. This is done. Now you understood what are the things that these people have taken. Yes, ma'am, we understood that there is a bacteriophage. There is two labeling substances we have taken. One is a P32 and one is a S35, right? Now what is going to be the first job, students? We are going to take an E. coli. Because we know bacteriophage is a virus that infects the bacteria. So we are going to take an E. coli cell. Okay, we are going to take an E. coli cell first. Now, okay, this E. coli cell is first going to be infected. Okay, first step is going to be what? Infection. What is infection? We are allowing the bacteriophage to infect this. So one bacteriophage is coming and it is going to be infected. But before we go to this step, there is one step that all of you need to know. Now I told you that labeling is going to happen. So we are going to indicate labeling with red color. Okay. So before I go to this, we do something called as labeling. Okay, labeling. Now what is labeling? I'll tell you. So first we take a bacteriophage. Okay, this is a bacteriophage. We'll just copy paste the same thing, so it becomes easier for us. Okay, this is our bacteriophage. Okay, now inside this, what do we have, students? We have the genetic material. Everything I'm drawing with white, so I can label it and I'll show you. Okay, you're also seeing. Now, first I am going to make sure that I radioactively label it. So first I am going to radioactively label it with sulfur. Now, if I am going to radioactively label with sulfur, which of the portion will be radioactively labeled? Can anyone tell me? If I am using sulfur, S35, for radioactively labeling it, will my protein get radioactively labeled or will my DNA get radioactively labeled? Quickly in the chats. Come on. Come on, come on. Interaction is important. Come on, quickly. And I hope all of you are marking your attendance as well. Yes. Excellent. Protein, ma'am. Okay. Protein. So, here if I am going to radioactively label my protein, so students, radioactive labeling I am showing with red, okay. I am, oh, this pen is going wherever it wants. Ooh, yeah, okay. My protein code got radioactively labeled, correct. My radioactive labeling happened with sulfur, so obviously my protein is going to get radioactively labeled. So when I use sulfur, protein got labeled. Now why am I doing labeling? So I want to know which is getting transformed into the cell. Why? Because E. coli is there. I am going to allow the E. coli to be infected with the bacteriophage. Whatever is entering into the cell is the person who is able to do transformation. If you do not enter into the cell, if you cannot change the mechanism of the cell, how will transformation happen? That is the thing, okay. Now this is one part. Here I am going to take what, students? I am going to take phosphorus. Okay, I am going to take P32. Phosphorus I am going to take. So here all of you know, if I am taking phosphorus, who is going to get radioactively labeled? My DNA. DNA is going to get radioactively labeled, right? So here we go. DNA gets radioactively labeled. Done. Easy. Easy. If I am taking phosphorus, DNA is labeled. Okay, DNA is labeled. So this is called as labeling. Now, very simple, okay. So I'm telling the conclusion of the experiment, then I will come by the steps. So this is your E. coli. This is your E. coli. This is the E. coli, no, students. If you spot, okay, very simple question I'm asking you. If you spot protein coat inside, okay, let's say that scenario doesn't happen, but let's say if you notice protein coat, okay, if you notice protein coat, what does it indicate? That okay, protein is in the outside, okay, or protein, wherever you are seeing the protein labeling, you will be noticing that okay, this protein is outside. And if you notice genetic material inside, like if you notice DNA inside, that means what? That means this is why we are doing labeling. This is why we are doing labeling. Otherwise, you will not be able to know where the protein is, where the DNA is. Understood? So, this is what your experiment is all about. Now, I will make you understand the steps involved in the experiment. Okay? Okay. The first step is going to be, students, first let's draw one E. coli. Okay, this is a E. coli. Little out of shape E. coli it is because it went through depression. So, okay. So, this is the E. coli. First, what am I going to do? First step for me to happen is going to be infection. How will I do infection, students? Ma'am, so simple, ma'am. You just let the bacteriophage do the job, right? So, if a bacteriophage is going to be near it, it is automatically going to attack this, right? It is automatically going to attack the cell, right? Now, another maybe, I think we will take it for until we finish starting of replication, okay. So, this bacteriophage is going to infect. So, students, it is automatically understood this as soon as a virus, okay, as soon as a virus infects a cell or infects the host, what is the first thing that it will do? It will release the genetic material. It will release the genetic material into the cell. It will release the genetic material into the cell. Correct? That's what it is going to do. So, when a bacteriophage affects the cell also, first thing this fellow is going to do is going to release the genetic material inside. Now, they are radioactively labeled or not, that is the second story. But first thing these people are going to do is that. So, the first process is infection, where the bacteriophage infects the E. coli. Second thing is going to be, students, blending. Why we do blending? Mainly we do blending so as to remove the protein coat from the outer surface, because this protein coat will be attached to the E. coli, no? To remove that we do blending. So, to remove protein coat, okay, which is attached to the E. coli cell, okay, we are going to do that. Last step is called as centrifugation. Last step is called as what? Centrifugation. Now, I want all of you to understand what is centrifugation. Students, simple centrifugation is, you take a test tube. Okay, this is your centrifugation tube. It will be little pointed at the end. Okay, this is your centrifugation tube. Okay, in the centrifugation tube, what is going to happen? This centrifugation tube, you are going to play it at a particular RPM. Huh? What, ma'am? Okay, so it is going to rotate. Students, there is going to be an equipment where we are going to do the rotation. Okay, so here one centrifugation tube, here one centrifugation tube, here one centrifugation tube, here one centrifugation tube will be kept. Okay, now all these tubes will be kept like this. Understood? Like this, the tubes will be kept, and we will rotate it at a certain RPM. Okay, certain RPM, that is at a certain speed, so as to get what? In this tube, you will be getting two regions. First is called a supernatant. All of you have played with mud, soil and water. How many of you have played? Soil and water, how many of you have played? Hey, mice is over, da. You're still stuck in mice, huh? Tell me. Students, tell me. Faster. Supernatant. So, if you have played with soil and water, you will mix the soil, you will pour water into it, and you will let it settle for some time. You will notice that the dirty water will come on the top and the soil will settle down. Something like that only. When at a certain speed we use this, we get a supernatant which is of the lightweight, and then we get a pellet. Okay, then we get a pellet like this. Okay, then we get a pellet like this, which is going to be at the bottom. Understood? So, this centrifugation is mainly done to separate the supernatant and the pellet. And obviously, genetic material is going to be in the pellet only because it's heavier, so it's going to be in the pellet. Now, we will come back to the experiment. Look at this experiment. All of you, open your NCERT in case the diagram is not completely visible. I think it's visible. So, look here. We have two bacteriophages. We have radioactive S35 labeled protein capsule, and we have radioactive phosphorus labeled DNA. Okay, so we are doing two types of infection. So first infection is happening where the proteins are radioactively labeled. Okay, and immediately I told you what is the first thing that the bacteriophage is going to do. It is going to release the genetic material. Can you see? Genetic material has been released into it. If you look here only one side, the protein coat is labeled and the protein coat is outside. It has not entered into the cell, right? Protein coat has not entered into the cell. So, the second process we do is called blending. Why? So as to remove the protein coat which is stuck to the outside of the cell. If you look at this place, you notice that the radioactively labeled DNA has entered into the cell, right? And then the third, what happens, students? We do centrifugation. Then we notice that there is no radioactivity detected in the cell. Why? Because the protein coat was labeled, and protein coat can only stay outside the cell. It cannot enter into the cell because only genetic material can enter into the cell. Only genetic material, like Gopika Ma'am's classes entered into your brain, like that genetic material can only enter into the cell. Okay. Now, here what they noticed, radioactivity detected in the cell. That means we noticed that P32 labeled radioactive substances was inside the cell. And then they noticed that radioactivity detected in the supernatant. That means sulfur 35, whatever was labeled, was noticed in the supernatant. And here what you noticed? No radioactivity in the supernatant. Why, students? Because we did not label the protein here. Is this understood? Quickly let me know in the chats. Is this understood, all of you? Is this understood or not? Faster, faster. Infection happened, right, where the bacteriophage infected the E. coli. Blending happened. That means where we, with a blender, we just removed the protein coat. Okay, and last, centrifugation happened, and we already know that radioactivity is going to be inside the cell. But here we still had to do the experiment. Okay, now we will go to NCERT. See, look here. This is a very, very important statement. Okay, the unequal proof, that means the one and only. Okay, we tell no unanimously, that means everyone agree to it. So the one and only proof that DNA is the genetic material came from the experiment of Alfred Hershey and Martha Chase in 1952. They worked with virus that infects bacteria called as bacteriophage. The bacteriophage attaches to the bacteria and its genetic material then enters the bacterial cell. This is what I told you. The bacterial cell treats the viral genetic material as if it is own. So virus nature is like that. Virus is like wherever I go, I am going to make it my own. Okay. So the bacterial cell treats the viral genetic material as its own and subsequently manufactures more viruses. This I'll teach you in HIV. When I teach you the life cycle of HIV, I'll teach you this. Hershey and Chase worked to discover whether it was a protein or DNA from virus that entered the bacteria. They grew some virus on a medium that contained what, students? Radioactive phosphor, and some on another medium that contained radioactive sulfur. Got it? Now, virus grown in the presence of radioactive phosphorus contained radioactive DNA. Why? Because phosphorus is only present in DNA and not in protein, not radioactive protein. Because DNA contains phosphorus but protein does not. Okay, protein does not. Similarly, virus grown on radioactive sulfur contained radioactive protein and not radioactive DNA. Okay, because DNA does not contain sulfur. Clear to all of you? Yes or no? Awesome. Okay, now radioactive phages were allowed to attach to E. coli bacteria. Then the infection proceeded. So first what happened? Infection proceeded. The viral coats were removed from the bacteria by agitating or else you could say simple word blending. Okay, blending. In the blender, the virus particles were separated from the bacteria by spinning them in centrifuge. Okay, bacteria which was infected with virus that had radioactive DNA were radioactive, indicating that DNA was the material that passed from the virus to the bacteria. Bacteria that were infected with virus that had radioactive proteins were not radioactive. Why? Because protein cannot enter into the cell. Right? Protein cannot enter into the cell. This indicates that protein did not enter the bacteria from the virus. DNA is therefore the material that passed from the virus to the bacteria. Any doubts, students? Any doubts? So this is what the experiment talks about. Clear to all of you, Hershey and Chase experiment? Yes or no? Yes or no? So Hershey and Chase experiment proved, okay, Hershey and Chase experiment proved DNA is the genetic material. Okay. Therefore, it was proved one second. What is the they use such fancy words, students. That DNA is the genetic material. Okay. So it was proved that unequally proved that DNA is the genetic material. Got it? Done? Any doubts? No, right? Okay. Now we have to study a little bit about DNA and RNA. I want 5 minutes to just fill my water bottle, so I will just resume back in 5 minutes. Okay, just 5 minutes, students. I'll just fill the water bottle and come. Don't go anywhere. I want someone to quickly give the attendance count as well. Okay. I forgot to fill, because of morning, all the problems. Science world, relax. Another 15-20 minutes class will be there. So much tension. Okay, come on. Let's continue. I just wanted 2 minutes to just go fill the water bottle. Okay. Now students, I want all of you to listen carefully. One more very important topic is, even though we keep telling that DNA is the genetic material, what is the main reason that DNA is the genetic material? Because even RNA we have. Why RNA is not considered as a genetic material? Questions is there. Okay? Yes? Understood? So, here first, okay. Students, there are some criteria for being the genetic material. Now, what do I mean by criteria? Like, let's say you want to stand for election, okay, or you want to vote. If you want to vote, that means you should be above 18 years and you should be a citizen here. These are few criteria that is given to you to be having the ability to vote or stand for any kind of election. Same thing happens in schools and colleges as well. If you want to be standing for the school captain or class rep or anything like that, there are some criteria that this much percentage you should have got, you should have been an active, attendance should be there. So these were the criteria for us in our school, okay. Now if you pass all that criteria, you can become the head leader, the head girl, the head boy, whatever you want to become. Now here also, if you have to be the genetic material, there are 4 criteria that you need to pass. That 4 criteria have to be gone through by your DNA and RNA, and then we decide who is going to be the genetic material. Okay. So the first criteria to be the genetic material is that these people should be chemically and structurally stable. What is it? They have to be chemically and structurally stable. Okay, all these notes will be available on Vedantu NEET English Telegram, and it will also be pinned to the comments. Okay, and if you want to connect with me, you can connect with me in Gopika Biology Official. So DNA, RNA. Now here comes the question. Why did we choose DNA is the question. See students, DNA is chemically and structurally stable because of mainly 2 reasons. First reason is the absence of the O group, okay, oxygen. Okay, I'll write oxygen only. Absence of oxygen in the second carbon. Correct? It's absence of oxygen in the second carbon. Yes, ma'am, I agree to you, because we know that this is your sugar, right? And in the sugar, we know in the second we have a hydrogen instead of an oxygen, right? In the second carbon, we have an hydrogen instead of oxygen. So because of that, one of the reason why DNA is chemically and structurally stable. And the second reason, okay, second reason is that they have thymine is present instead of uracil. What is present? Thymine is present instead of uracil. These are the 2 reasons why DNA becomes more chemically and structurally stable compared to RNA. But on the other hand, students, RNA, what happens? RNA sadly has presence, okay, presence of oxygen. Okay, I think I have to shift it a little. 1 minute. One second, students. Yeah, okay. So here, presence of, here what is happening in RNA? Presence of oxygen where in the 2' carbon is making it highly reactive. Okay, this oxygen, students, is a problem. It becomes highly reactive. Highly reactive. Okay, because of the presence of the carbon, it becomes highly reactive, leading to more problems. Okay, making it less chemically stable. Now, second thing, the problem is that these people, okay, these people, if you want me to draw here, I'll draw that also. Okay, they have an OH. Yes. Students, these people have one more problem. What is that? Presence of uracil. Okay, presence of uracil instead of thymine. Presence of uracil instead of thymine. This is also making them very unstable. This is also making them very, very unstable. Clear? Done? Okay, so first criteria is done. Okay, we'll go one by one, because last time, last time they had asked in statement based question from here. So that's why it's very important for you to know the reasons well. Thymine is present instead of uracil. Okay, these two reasons are mentioned in your NCERT, so we stick to these two reasons itself. Okay, now here, okay, next one, okay, here, let's go and try and understand. Students, the next reason is going to be replication. Replication. So, one more criteria is they should be able to do self-replication. Okay, they should form their replica. They should be able to form their replica. Got it? Yes or no? So, if they are not able to form their replica, okay, Nobita, yes, new to the class, awesome. So, if they are not able to form their own replica, then we do not consider them as a genetic material. But luckily here, DNA as well as RNA can form their own replica. They can both form their own replica. So, there is no problem here. Okay, there is no problem here. Second, third one is that they mutate. Okay, mutation, slow mutation. Slow mutation. What is slow mutation, students? Do you want to sleep like a human and wake up like a penguin? No, ma'am, I don't want to do that, right? So, slow mutation, students, DNA mutates very, very slowly. But on the other hand, RNA mutates very fast. So, if mutation happens very fast, then we cannot have that as a genetic material. We cannot have that as a genetic material because it's going to cause problems. So, slow mutation is followed by your DNA, okay? Then the last one, it should be able to express itself in Mendelian characters. Express itself in Mendelian characters. Students, they will be able to express themselves, yes, and both are going to express themselves. So, if you look at an average, okay, if you look at an average, and even we write the last point here also, so that you know totaling who is going to win, chemically and structurally stable. Who is going to win? Who is going to win this race? Come on, tell me who is going to win the race? Ma'am, it is going to be DNA. Why? Because DNA has got all the 4 done, right? They can do replication, they can form their own copies. So, here remember, they have to form their own copies, okay, or we can say replica. Okay, if they are not able to form their own copies, then they cannot become a genetic material. So, DNA and RNA can form their own copies. Slow mutation, DNA does slow mutation, but RNA does fast mutation. Express itself in Mendelian characters, yes, both of them are expressing. Chemically and structurally stable, RNA is not chemically and structurally stable. So, who is going to be the win-win? DNA is going to be the win-win. Done? So that is your, DNA versus RNA and the criteria to become the genetic material. Any doubts here? Please let me know in the chats. And if all of you understood, quickly put thumbs up in the chats. Okay? When will I ever draw a straight line? It's like one dream come true. And students, do not forget about filling the form. Those who joined late, okay, we will be coming very soon to Chennai and Hyderabad because that two cities have got the maximum votes. Okay, I'm so sorry for other people. But if you're from Hyderabad and Chennai and who has still not filled the form, quickly go and fill the form. It's a good available for DNA batch. So when you're taking a batch at 4,000, 5,000, we're also giving you the book as well. We're also giving you Tattva as well. So it's like a big combo that you're getting. It's not just classes. Okay, done? Done. Now, look here. Okay, I'll just read it from here only, students. I think this is better. So, here we go. Properties of genetic material, DNA versus RNA. Okay, so look here. From the foregoing discussion, it is clear that the debate between protein versus RNA as a genetic material was equally resolved with Hershey and Chase experiment. It became an established fact that DNA that acts as a genetic material, however, it subsequently become clear that in some viruses, RNA is the genetic material. So, the tug of war between protein and DNA becoming a genetic material was completely over. But the next tug of war was there between RNA and DNA being the genetic material. Okay? Right. So, now look here. For example, tobacco mosaic virus or your bacteriophage, etc. have RNA as the genetic material. Answer to some of the questions such as why DNA is predominant genetic material whereas RNA performs dynamic functions of what and all, acting as a messenger, okay, acting as a messenger, adapter, and has to be found in difference between chemical structures of two nucleic acids. Okay. Gopika Biology, that's my ID. Okay. Now, a molecule that can act as a genetic material must fulfill the following criteria. What are the criteria? It should be able to generate its replica. That means they should be able to undergo replication. It should be stable chemically and structurally. It should provide the scope for slow changes. That means mutation has to be slow, which is required for evolution. It should be able to express itself in the form of Mendelian characters. These are the 4 criteria. Now, okay, now let's see if they are able to do it. Now, if one examines each requirement one by one, because of the rule of base pairing and complementative, both the nucleic acid, that is DNA and RNA, have the ability to direct their duplication. That means they both can do replications. The other molecules in the living system, such as protein, fails to fulfill this criteria. They cannot, okay, fulfill the criteria. The genetic material should be stable enough to not change with different stages of our life cycle. Now, when we are child, when we are adult, we do not want our genetic material to keep changing, right? We want it to be constant. We want it to go through small changes, right? Yes? Okay. Hi, Hershey. Nandini, the votes are very less on Telangana. So, we told you, the highest voted cities will be the only place we can come because difficult to travel to all the cities, right? So, yes. But if any of you can come to Hyderabad, well and good, please try to make it. If it is like a 1 hour journey, please try and come because we'll be very happy and you are going to be, and it is in the evening. So, you have enough time to travel and reach. After college also you can come. It's only Saturday, right? Saturday and Sunday. So, you can come aram se after, like with your parents, you can travel and come if it is close by. Like if it's a small train service is there where you can travel. I'm not sure how it happens. But if there is a small transport service is there, please do come. Okay. Now, look here. The genetic material should be stable enough to not change in different stages of life cycle, age, or with changes in physiology of the organism. Just because I do something outside, okay, I do a facial treatment, do I want my genetic material to be affected? No. Okay, some person went through a fire, okay, and got half of his body burned. Do you want his genetic material to be damaged? No. So, we want someone who is going to be stable no matter whatever happens in the outside. Okay, no matter whatever happens in the outside. Okay, so stability as one of the properties of genetic material was very evident in Griffith's transforming principle itself, that heat which killed the bacteria, okay, heat which killed the bacteria at least did not destroy some of the properties of the genetic material. Yes or no? See, it would have damaged the genetic material, but some properties stayed intact. Okay, this is now can be easily explained in the light of the DNA that the two strands being complementary, if separated by heating, come together when appropriate conditions are provided. What are they talking about, students? See, this is your DNA strand. This is one strand, this is one strand. They both are wound around each other, right? Now, if I am separating it by heating it, oh, let's say, okay, I am heating this strand, okay, and both the strands got separated. Okay, this is one strand and this is one strand. 5 dash to 3 dash, 3 dash to 5 dash. Now, after the condition becomes favorable, these both will have high tendency to come back together. This will have high tendency to come back together. Why? Because they both are complementary, right? Yes. Now, when appropriate condition. Further, 2OH group present at every nucleotide in an RNA is a reactive group and makes RNA liable and easily degradable. That means this fellow has become problematic. His life has become problematic because of the OH group. Okay, right? Yes? I'm in Delhi, Haryana, Kashmir. Oh, you people are so far. But it's okay. One day we become big, we will come to all the cities. Okay? So you people have to make that happen. RNA is also now known to be a catalytic, hence reactive. Therefore, DNA is chemically less reactive and structurally more stable when compared to RNA. Therefore, among the two nucleic acids, the DNA is a better genetic material. Got it? Better genetic material. In fact, the presence of thymine, see, presence of thymine at the place of uracil also confers additional stability to DNA. Detailed discussion about this, you will be understanding the process of repair in DNA. You will not study now in higher classes. Okay, both DNA and both DNA and RNA are able to mutate. In fact, RNA being unstable mutate at a faster rate. Consequently, virus having RNA genome and having shorter lifespan mutate and evolve faster. RNA can directly code for the synthesis of protein, hence can easily express the characters. DNA, however, is dependent on RNA for synthesis of protein. The protein synthesizing machinery has evolved around RNA. The above discussion indicates that both RNA and DNA can function as a genetic material, but RNA is more stable and it is preferred for storage of genetic information. Clear? Yes or no? Yes or no? Nandini, go wash your face. For the transmission of genetic information, RNA is better. So, this is your NCERT line. Students, in NCERT, one thing they mentioned. I'll write it here, okay? I'm writing it in a different color, so that means it's a different concept, okay? So, let me put it in a box and just write it here. See here. So, they are saying that RNA, okay, so there's one topic called as central dogma. Okay, there's one topic called as central dogma. That means, students, our entire molecular biology runs on the central dogma. What is central dogma? You people already know about it. It is very simple that DNA can form RNA and RNA can synthesize proteins. Now, if you look here, okay, if you look at this, they are saying that DNA is dependent on RNA, right? DNA is dependent on RNA to make protein. Do you agree or do you disagree? DNA is dependent on RNA to make protein because DNA cannot directly make the protein. But if you see here, RNA can directly form protein. Yes, ma'am, RNA can directly form protein. So, here what is happening? DNA is dependent on RNA, but RNA can directly form protein. So, here they are saying that RNA can synthesize protein directly. That means without the help of DNA. But DNA cannot, I write synthesize in short form, synthesize protein without RNA. That is what they say. But still, DNA is the most stable material. Okay? DNA is the stable material. Done? So, this is what I wanted to explain to all of you. So, I think we will stop it here. Okay? This I made you understand now. Just RNA world we will do and then I will stop. Okay? I'll take replication in the next class. Okay? Got it? Because it's lunch time and I know most of you will be hungry. So, here RNA world means what, students? Other than RNA being genetic material, see, RNA is a genetic material, no doubt. RNA is genetic material, but for very, very, very specific organisms, right? RNA is genetic material for here as of now tobacco mosaic virus, and for what? For your bacteriophage. Okay, right, these are the people who is going to have RNA as the genetic materials. Okay, now other than that, what are the functions that RNA does? RNA acts as a messenger. Yes or no? RNA acts as a messenger. RNA acts as an adapter, which is one of their best functions. Okay, it acts as an adapter, and this is the one who is going to help us with translation. Okay, translation. Then RNA also has catalytic functions. Okay, catalytic functions. So RNA, if you see, even though it is not a genetic material in 99% of organisms, it is only a genetic material in few viruses. It has other main jobs of messenger, adapter, catalytic function. So here what do we understand? We understand that DNA is dependent on RNA, not just for one process, students, for many processes. So we can also say that people say that RNA was the first genetic material. RNA was evolved first. So should we respect RNA? Yes, ma'am, we have to respect RNA. Okay. Evening is the 11th classes will be on evening, 12th will be, I'll see students, I'll try to alternate. Okay. Okay, done. So this is about your RNA world. Okay. Look here. From foregoing discussion, an immediate question becomes evident: which is the first genetic material? It shall be discussed in detail in the chapter on chemical evolution. But briefly, we shall highlight some facts. RNA was the first genetic material, and there is now enough evidence to suggest that essential life process, that is metabolism, translation, splicing, etc., evolved around RNA. You will understand later, don't worry. RNA used to act as a genetic material as well as a catalyst. There are some important biochemical reactions in living system which are catalyzed by RNA, not by protein. But RNA, being a catalyst, was reactive and hence unstable. Therefore, DNA has evolved from RNA with chemical modification that make it more stable. DNA is being double-stranded and having complementary strand further resists the changes by the evolving repair. So here what is happening? RNA was evolved first, but because of their catalytic nature, they became more unstable. Okay, they became more what? Unstable. This much can you remember? Okay, if in case any questions come, you should be in a position to tell this. Okay, yes, ribozymes. Okay, understood? Now, last concept for the day, central dogma, and after that, we will stop. Central dogma. Okay. Now, students, central dogma, as I told you, it is the entire rule, okay, of your molecular biology. That is, DNA to RNA to RNA, RNA to protein. This is what the whole chapter is talking about. This is what the whole chapter is talking about. DNA will form mRNA, mRNA will form protein. Now, DNA forms mRNA. Students, the process is called transcription because you are transcribing something. Okay, transcription is going to happen. mRNA to protein is going to be translation because you are translating something and making the protein synthesis go. Now, one more very, very important process is going to be DNA able to form its own copies. DNA is able to form its own copies. Now DNA is able to form its own copies means what? It will be able to form replicas. That process is called as replication. That process is called as what? Replication. So now in our further classes you are going to learn replication, transcription and translation. Clear to all of you? Any doubts so far? Please let me know. Understood this much, right? So if DNA is going to do replication, that means it is going to make its own copies. We call that process as replication, and DNA to mRNA we call it transcription, mRNA to protein we call it translation. Okay. Now in between this there is going to be a lot of things that you are going to study, like RNA processing, splicing, genetic code, that we will come slowly. Okay, that we will come slowly. Okay, awesome. Awesome. Understood? Now, okay. Can you remember this word? DNA replicates in a semi-conservative manner. DNA replicates in a semi-conservative manner. This is very, very, very important. Okay, our DNA replicates in a semi-conservative manner. Clear? Now, to teach you semi-conservative manner, I have a small trick, okay? Small trick, I would say, but I don't know if it will fit in here. Okay, just look here, students. Just looking at the color, you tell me if you are able to understand something, okay? I'll first draw, then I will tell you. Okay, this is one strand. Can you see blue color is one strand, right? Now this blue color strand, students, I call it the parent DNA. I call it what? The parent DNA. Now what do I mean by parent DNA? That means, example, now let's say I have this book. Okay. Okay, I have this book. I need one student's name. Whose name do I take? Okay, let's take Nandini Oli. So this book I have. Nandini came and asked me this book. I told, okay Nandini, you take the book. She took the book. Now this becomes the parent copy, right? This book I have given to Nandini, so this becomes the parent copy. Now Nandini takes the Xerox and comes, right? Nandini takes the Xerox and come, and let's say this is the Xerox that Nandini has took. Now this becomes the new copy, right? This becomes the new copy, daughter copy we can say, or we can say the Xerox copy, right? So this was the original parent copy. From the parent copy she got a Xerox, so this becomes the new formed. So can I call this the template? Can I call this the template? If I am telling you that, okay, let's say I am telling you I want your presentation to be same template, and I am giving you this template. You will copy paste this to a new copy. So this becomes your template and this becomes your new copy. Understood all of you? So here also, students, this becomes your parent DNA, the original copy. I have separated the parent strands, okay? I have separated the parent strands, okay? Let's say one is from 5' to 3', one is from 3' to 5', okay? I have separated the parent strand, and after separation, okay, after separation, I am letting them form copies, okay? So one copy formed, okay, 3' to 5'. One more copy formed, 5' to 3', correct? Now this becomes the new copy, yes or no? This becomes the new one. So here I am writing P, parent copy, and N for daughter DNA or new copy. Again N and P, right? This is what is happening. Now what do you expect? After this, what do you expect? Ma'am, I expect that these people come together. Right, 1 second. Okay. Yeah. So, what do I expect, students, after this? I expect that these people come back together, right? So, I am saying that, okay, parents are going to come back together. Okay, I took the parent copy from you, and the daughter DNAs also are going to come back together because they are form complementary. Now look here, again this is your parent DNA, This is your newly formed strand DNA. This is the new one. Right. Now, this kind of a technique, what you are seeing here, this kind of a technique is called as conservative. This kind of a technique is called as conservative method. Conservative method means what? After parents give the DNA, we form the copies, parents go back, daughters go back. This is the conservative method. But our DNA replicates in a semi-conservative manner. Can you all remember this much? Can you all remember this much? If you can remember this much, I'll tell you one thing. Interesting, students? Yes or no? With the same thing, I'll teach you one more thing. Okay, so yeah. So this is 2 boxes. Okay, I want you to identify, clearly see the box. Okay, now here what I'm going to do, this last step I'm going to remove. Students, normally this is what was supposed to happen. One newly formed strand. Understood? So instead of like this, if you have any doubts, conservative means what? Let's say parents are coming to solve our problems. Okay, they solve the problems and then they go back together. That means they're like, okay, whatever, we siblings will become together. Here what is happening? Parents came to solve the issue. Okay, let's say father took elder daughter's side and mother took younger daughter's side. During the fight, suddenly father tells something about mother or tells like, oh, she's like you. And mother gets triggered and mother starts fighting. Yeah, she's also like you. And then what happened? Father also fights, mother also fights, daughters also fight. And then it becomes like father and daughter becomes one gang, mother and daughter becomes one gang. Understood? So it's something like that, semi-conservative. Okay, one parent strand and that is one old strand, one new strand, one old strand, one new strand, or one heavy isotope, low isotope. So we will teach you, I'll teach you the experiment. Please give me a thumbs up if you understood this. Because with this only I'll be continuing the next class. So if you do not understand this, next class will be difficult. Understood? Understood? So we are going to get hybrids here. Okay? We are going to get hybrid strands here. Because we are getting one parent and one newly formed strand, or we can say daughter strand also, but yeah, N. Okay, one newly formed strand. So we are getting hybrid. Yes, ma'am. Perfect. Awesome. So this is what replication is going to be. So understand now, our DNA replicates in a semi-conservative method. Next class, I'll teach you how the replication happens, and I'll teach you what is replication, at which stages it is happening, and where is it happening in eukaryotes, prokaryotes. So that will be more detail of a class. Okay, so I'll do next class replication and transcription most probably, and if possible genetic code I'll do. So let's see how the class goes. Okay, next class will be a little more longer class. So be prepared. Okay, be prepared. I think I'll try to take it in one of the evening slots so that you people will not feel the burden. Okay, of that, ma'am, you're teaching so much, ma'am. Okay. Right, so much teaching is going on, ma'am, so that feeling will not come. So next class I'll teach you replication. Okay, replication I'll teach you in detail. So students, today is everything you understood, yes or no? Did you understand everything? Central dogma, RNA world, okay, DNA versus RNA, then you understood Hershey and Chase experiment, then what did we do? Avery, MacLeod and McCarthy's experiment. This also understood, I hope. Okay, this doesn't have heading only, 1 second. Because they removed the heading, no? Done? Okay. Then what did we learn today? We studied Avery, MacLeod, Griffith's experiment, right? And we just understood, and this is the most important thing. Okay, 5th and 6th July. Okay. Okay, sure, I'll put the notes. Students, last class, see, look here. If you don't know where the notes will be there, no, just go here. See, now you're, go to Vedantu NEET English. Let's just go to any of the live. Okay, let's say last class I took Animal Kingdom. Okay, if you go here, where did it go? Live. Okay. Yeah, let's go to Animal Kingdom. See, students, session PDF link will be given. Click on this, enter your phone number, and directly you will be able to download the PDF. Understood? Every class there will be notes. Every class there will be notes, and it will also be pinned to the comments. So don't wait for me to share it on the Telegram. Directly here it will be there. In the mid of the year, like in another 2 months, I will share all the notes together on my Telegram channel. Okay, so that time you can join and download it directly. Right now, you do this. Okay, and I want all of you to let me know in the comments if you understood. Last class I was very disappointed. So much hard work into, you know, making the whole thing. And students, please understand, teachers also make notes. Okay, this is the notes that I have made for you, so I can share it on the Telegram channel for all of you. So can you see different colors? Okay, everything is done for you. This whole notes I will be sharing with you. So I want all of you to take that 5-10 minutes, not even 5-10 minutes, 2-3 minutes to just tell me how the class was. It's not just to make me happy, it also gives another student confidence to open the class, right? Because we are all small teachers and we all start very small. So you people should make us, you know, help us so that our classes can go to more other students. Okay? Yes? Done? Clear? So this, after the session notes will come. Before the session only notes will come. What I'm machine or what? First I will write, then I will do class. You people, where your brains you people have kept in come, I don't know. Okay, so this, yes, quickly smash the like button as well. I want to see more likes on the video. And see, every class like that notes will be there. Okay, molluscas is there here. DNA, genetic material. DNA batch, you have one more chance to get in. So students, you're going to get 300 plus live sessions by master teachers. And you're going to get instant class doubt solving and 40 plus part and full test. Okay, and you're also going to get Tattva book PDF. And please understand that here right now the price, there is a drop. Okay, price there is a drop. So 3,999 for 1 year, 4,999. This is with the flashcards. I would suggest you go for this because flashcards are the best thing. Okay, I use flashcards whenever, like, you know, when you're traveling. When you're traveling, keep the flashcards. Okay, you want homework? Why not? I'll give you questions. Okay, wait. Okay, here only. Take down the questions and give me the answer in the chat box. Okay, in the comments. Okay, first question for all of you. What was used to label DNA in Hershey and Chase experiment? Okay, what was used to label DNA in Hershey and Chase experiment? I want all these answers in the comments. Okay, don't answer now. Comments. Okay, first one. Second one. Which use of which enzyme inhibited transformation in Avery, McLeod and McCarthy's experiment? Okay, these two. These two, do it as homework. Okay, homework. Do this two as homework. What was used to label DNA in Hershey and Chase experiment? Use of which enzyme inhibited transformation? Okay, both should come on the comments. Clear? 1 and 2. 1 and 2 questions should come in the comments. Clear? This is your homework. I hope all of you are able to, you know, make a note of this. What was used to label DNA? Important. What was used to label DNA in Hershey and Chase experiment? You can write the short form or you can write the full form. Use of which enzyme inhibited transformation? Use of which enzyme inhibited transformation? Clear? Yes or no? Use of which enzyme inhibited transformation? Yes? Awesome. Awesome. Okay. Thank you so much, students. Homework I should see. Who is the one who asked homework? Nandini, I have to see your homework also. Everyone's homework should be there in the chats. Okay, students. Take care. Bye-bye. Notes will be shared. Don't worry. Okay? Notes will be shared. Don't worry. Thank you so much. Take care. Bye-bye. And do not forget to fill the form if you haven't yet.